Welcome to this explainer. Today, we're taking a fascinating journey into the microscopic world to explore the ultimate defensive armor of bacteria, the cell envelope. Have you ever wondered how these tiny single-celled organisms manage to survive incredibly harsh environments? Or, for that matter, how our most powerful medicines actually defeat them? Well, you're absolutely in the right place. By the end of this, you're going to have a crystal clear understanding of one of the most structurally vital and targeted features in all of microbiology. Let's get into it. Okay, let's dive into this. To really wrap our heads around how a bacterium protects itself, I want you to consider this quote from historian Barbara W. Tuchman describing a medieval knight. The basics were a suit of plate armor, all worn over a hauberk or shirt of chainmail. Chainmail covered the neck, elbows, and other joints. Now, this isn't just a fun history fact. It actually turns out to be the absolute perfect metaphor for our topic today. Because, as we're about to see, a bacterium operating inside the human body is essentially a microscopic knight. It's suited up in its own precise version of chainmail and plate armor, perfectly engineered for battle. So here's our roadmap for today. First, we'll look at the bacterial armor itself. Second, we'll compare gram-positive versus gram-negative. Third, we'll go inside the peptidoglycan. Fourth, we'll look at the role of PBPs. And finally, we'll wrap up with why this all matters. We're going to break down these complex concepts piece by piece, so don't worry, you've got this. All right, part 1, the bacterial armor, a cell's ultimate shield. Every bacterium out there is surrounded by what we call the cell envelope. Think of this as its primary line of defense. Now, just like our own human cells, a bacterium has an inner cytoplasmic membrane that keeps its internal components beautifully organized. But here's the catch. Unlike human cells, which generally get to bathe in a nice, highly regulated isotonic environment, bacteria live out in the wild. Or worse, in the deeply hostile environment of the human immune system. So the cell envelope is this tough, rigid outer layer sitting right on top of that membrane. It gives the bacterium its characteristic shape, whether that's a rod, a sphere, or a little corkscrew, and critically, it protects the organism from severe mechanical and osmotic stress. Literally, without this envelope, a bacterium taking in water would just swell up like a water balloon and burst. Moving right along to part 2, gram-positive versus gram-negative. Let's look at the two major styles of this microscopic armor. Now, what's really interesting about this slide is what's happening on the top versus the bottom. If you look closely at the top half, you can visually trace that thick blue layer. That is the cell wall of a gram-positive bacterium, and it's sitting directly on top of the inner membrane. But now compare that to the bottom half. The gram-negative bacterium has a much thinner blue cell wall, and it's sandwiched beneath a thin purple line sitting on the very outside. That purple line represents an entirely secondary lipid membrane. Let's contrast these two types a bit more methodically. Gram-positive bacteria rely on a massive, thick cell wall. When scientists use a gram stain in the lab, this robust wall holds onto the dye and stains a deep blue or purple. Gram-negative bacteria, on the other hand, have a much thinner cell wall, which causes them to stain pink. But hey, do not let that thin wall fool you. They totally make up for it by adding that extra outer membrane we just looked at. Going back to our medieval knight metaphor, if the cell wall is the chainmail, gram-negative bacteria are wearing a heavy suit of plate armor directly over their chainmail. And to make things even more formidable, that outer plate armor contains lipopolysaccharide, or LPS for short. That's a molecule that triggers a massive and often toxic inflammatory response in humans. It's serious stuff. Now, because gram-negative bacteria have this highly complex double membrane system, we really need to define a specific term here, the periplasmic space. Imagine the gap between a knight's innermost tunic and their outermost steel breastplate. Got that image? Well, in a gram-negative bacterium, the periplasmic space is that distinct gap between the inner cytoplasmic membrane and the outer membrane. And sitting right inside the space, just floating between the two lipid layers, is where you're going to find that thin cell wall we just talked about. But think about this. Here's a super interesting logical problem. If a gram-negative bacterium is completely sealed inside a solid outer membrane of plate armor, how does it actually eat? Like, how does it interact with the world at all? Well, even plate armor needs breathing holes, right? Enter the porins. Porins are these highly selective protein channels located right within that gram-negative outer membrane. They basically act as vital gates, allowing specific required compounds, like molecules used for metabolism, to readily pass through into the bacterium. All the while, they keep dangerous molecular threats, like many types of antibiotics, locked entirely out. Pretty clever design, if you ask me. Okay, part 3. Inside the peptidoglycan. We are now zooming in much closer, past the outer membrane, and looking straight into the cell wall itself, the bacterial chainmail. The cell wall itself is made of a really complex substance called peptidoglycan. And to understand its structural brilliance, we just need to look at its basic building blocks. It consists of long polymers made of two alternating sugars, N-acetylglucosamine, which we just call NAGA for short, and N-acetylmuric acid, which we call NAMA, so NAGA and NAMA. These sugar molecules simply link together to form long, repetitive chains. And hanging right off of these chains are short little tails called peptide side chains.
So the crucial point is this. If the cell wall were made of just those NAGA and NAMA sugar polymers alone, it would actually be incredibly weak and flimsy. The true strength of the bacterial cell wall doesn't come from the sugar chains themselves, but from how they are chemically connected. Those little peptide side chains we just mentioned, they reach out and form physical crosslinks with the side chains of neighboring polymers. It is literally the exact same mechanical principle as medieval chainmail. Individual metal loops are easily bent, but when you physically cross-link them all together into a tight mesh, boom, you get an incredibly tough, impenetrable layer of armor.
Which brings us to part 4, the role of PBPs, microscopic blacksmiths. Let's meet the workers responsible for forging this chainmail. And this brilliantly illustrates exactly how the bacterium builds its chainmail. Notice the long, alternating chains of the green NAGA and NAMA ovals. But look closely at that large orange circle labeled PBP. That stands for penicillin-binding protein. These PBPs are the specialized bacterial enzymes that act as microscopic blacksmiths. They actively grab on to those peptide side chains and forge the physical connections that hold the entire cell wall together. Let's walk through the exact mechanical function of these penicillin-binding proteins step by step. Step 1, the PBP blacksmith recognizes the very end of a peptide side chain, specifically seeking out two terminal amino acids called D-alanine. Step 2, in many bacteria, the PBP then helps form a structural bridge made out of molecules called glycine residues. Step 3, the PBP physically cross-links the two side chains together. It's amazing. With every single bond they forge, the bacterial armor becomes stronger and more rigid. Let's move to and see how this builds in real time. Looking right at the bottom of the diagram, we can see the orange PBP actively catalyzing that exact alanine-alanine peptide bond. This is the precise moment of construction, folks. The enzyme locks onto the chemical structure of the cell wall components and perfectly fuse them together. Without these PBPs working tirelessly around the clock, the bacterium simply cannot maintain its chainmail. The armor would fall apart under the intense osmotic pressure, and the bacterium would just die. And finally, part 5, why this matters. Let's tie all this dense microscopic biology back to human medicine. Here is the absolutely incredible takeaway from everything we've explored today. Remember how we emphasized that human cells have a cytoplasmic membrane, but they do not have a cell wall? We don't make peptidoglycan, we don't utilize NAGA or NAMA, and we definitely don't have PBPs. This means that the bacterium's greatest strength, its highly specialized protective armor, is actually its fatal flaw. Medical science realized that if we can engineer drugs that target this unique cell wall machinery, we can destroy the bacteria without harming human cells at all. It's a total game changer. And that is exactly what the most famous antibiotics in history, like penicillin and other beta-lactams, do. They act like a specialized weapon, kind of like a crossbow, designed to jam up those PBP blacksmiths, totally stopping them from cross-linking the chainmail and causing the bacterial armor to literally burst open. But how exactly do these antibiotic crossbows trick the PBPs? Well, that is a spectacular story of molecular mimicry and definitely a question for next time. Thanks for joining this explainer, and I'll catch you on the next one.